based on what makes sense topically. So Sam Jett from University of Oklahoma is the first talk, talking about biaxial mechanical testing, constitutive model development. What's wrong with me? Atrioventricular valve leaflets.
Well, thanks, Dr. Bergen, for that introduction. Yeah, we're from the Biomechanics and Biomaterials Design Lab at the University of Oklahoma. And we'll start off with an outline to the presentation I'm going to give today. So it'll start off with kind of an introduction to heart valves and leading to the motivation for this study. And then we'll move into the biaxial mechanical testing that we've done and the constitutive model development to kind of relate, to be able to more fully capture that experimental data and put that to a model, and then the summary of this study and the clinical impacts. So we'll start off with kind of an introduction to the heart function. So here you can see 4 main chambers within the heart, and we'll start looking at the right side here, which I realize is put on the left, but it's kind of from a morphological view, it's the right side. So what happens here is deoxygenated blood flows into the right atrium, and it flows in through the superior and inferior vena cava, and then it passes from this right atrium into the right ventricle. And in that process, it flows through the tricuspid valve. Once the deoxygenated blood is within this right ventricle, the right ventricle actually contracts, and you can see all this myocardial tissue that allows that to happen. And then the blood is forced out of the heart and into the lungs. So you can see that process here. And from the lungs, we'll then consider the left side of the heart where that oxygenated blood actually returns. So it'll return from the pulmonary vein into the left atrium. So you can see that oxygenated blood comes here, and then it passes through the mitral valve here. These are known as the 2 atrioventricular valves, mitral and tricuspid, and that's because they sit between those atria and the ventricles. But this mitral valve allows the blood to pass into the left ventricle, and the left ventricle actually contracts once again using the musculature. And actually, there's a lot more musculature in the left ventricle because that has to push the blood into the aorta and through the rest of the body versus just the lungs. So actually, we'll look at an overhead view after this, just to kind of see a little more about the anatomy of these valves. So you can see the 2 leaflets of the mitral valve right here, the anterior and the posterior, and then the 3 leaflets of the tricuspid valve. And we'll call these TVAL, TVSL, TVPL for the rest of the presentation. But you'll also notice some interesting features, like right here, the chordae tendini. And these are small tendons that actually allow for the valve to function properly. And what they do is they anchor those leaflets. As the leaflets, what's known as coapt, the valve closes and it stops the blood flow. And you'll also notice these rings around the valves. These are known as the valvular annuli, and they basically confirm the geometry and enclose that geometry. And so we'll actually look at some conditions that can affect these heart valves. But principally, these leaflets are the focus of this study. So here you can see a healthy and regurgitant, both mitral and tricuspid valves. And the key to notice here is that in the healthy case, all the blood is pushed out of this ventricle and none escapes to the atrium. Whereas in the regurgitant case, you get this blood flow back into the atrium in both mitral and tricuspid valves. So this is basically just an improper closing of the valve. And there's a couple different ways to treat this because it can lead to degraded heart function, actually to heart failure eventually. But one of the ways to treat that is an annuloplasty ring repair. These have gotten more common in recent years, but if there are no damaged components of the heart valve, this is a good way to do it. And so one of the issues this fixes is that when this annulus dilates and the leaflets are no longer able to meet in the middle, what happens is you have an open heart surgery and the surgeon suture on this annuloplasty ring, and that allows the geometry to be constrained and the valve to close fully. And another option for that is replacement heart valves. Here you can see actually a transcatheter heart valve, as well as a bioprosthetic heart valve that's not delivered via a catheter, it's delivered through open heart surgery, as well as a mechanical valve on the far right. But really, none of these options are ideal. They have issues with regards to longevity. Generally, these repairs last for 6 to 8 years, or sometimes slightly even shorter, and that's because these valves become degraded and they lose their efficacy. It's also very difficult to match the proper treatment to the patient. It's hard to get proper imaging and a full understanding of what's going on inside the patient's heart prior to surgery, and so it's difficult to choose what would be the optimum approach. So this study is actually going to focus on quantifying the mechanical properties of these mitral and tricuspid valve leaflets to better understand how that valve functions and to lead to some actually very interesting potential fixes for some of those issues for heart valve treatment. So we look at these mitral and tricuspid valve leaflets, and specifically we want to understand how they respond to their loading in their physiological state. And actually to do that, we do some experimental testing. And so that's what I'm going to move into in this next in this next part. So we'll talk about the biaxial mechanical testing of heart valves. And our first step in that is the dissection of the heart valve leaflets. So you can see right here the mitral and tricuspid valve leaflets. In this study, we use porcine heart, so that's what it's shown here. What we do is we are able to make cuts longitudinally along the heart and open it up, as well as excise these leaflets. So here you can see a TVAL just laid out. And our next step is to prepare the leaflet specimen for testing. So in that process, we make sure to clarify this radial and circumferential direction. You'll hear me say anisotropic a lot later, and what that means is a directionally dependent response. So these leaflets have that anisotropic property. So we make sure to clarify the radial and circumferential directions, and then we mount it onto our tester and apply these 4 fiducial markers. I'll discuss the reason for that in a moment. But once the tissue's mounted, we're able to apply these biaxial mechanical testing protocols. And what these are basically ratios between the applied force in the x and y direction, which actually correspond to the radial direction and the circumferential direction. But what we start off with is a preconditioning protocol. And what this is you can basically imagine stretching a balloon. You know how you have to kind of warm it up so that it can expand fully. So what we're doing is taking the tissue in this excised state, and we're essentially returning its mechanics to their physiological state through that stretching. And then we apply a simple 1-to-1. We call this equal biaxial loading ratios. And we also apply these 0.75 to 1, 1 to 0.75, 0.5 to 1, and 1 to 0.5. And what these non-equal biaxial loading ratios do is allow us to kind of fully capture the loading state of that leaflet in vivo, how that leaflet would be loaded in vivo, because we don't know exactly whether it's going to be loaded equally in each dimension. So once we've got our testing completed, we have to process our data. And the first step in that is this optical strain tracking of these markers. So this optical strain tracking allows us to obtain this large matrix of coordinates for these 4 nodes. You can see how they deform here. And then basically from those coordinates, which we obtain through digital image correlation techniques, we're able to do shape function analysis and to back out the deformation gradient. And you can see that the principal stretches of the deformation gradient are actually the principal stretches of the tissue. So after that, we're able to compute specimen stresses just based on the applied loads. We have load cells in each arm of the biaxial mechanical tester, and we also note the original geometries prior to loading. And then from that data, we're able to produce these sort of graphs. So you'll notice that there's kind of 2 mechanical response curves here, and that might seem a little counterintuitive. That's because this material is so anisotropic. So you can see how the radial stretch is much more than the circumferential stretch. You'll notice this is under equal biaxial loading, but that this tissue has just a very pronounced anisotropy within it. And so we capture that through the biaxial testing. So as far as our average results, we tested n equals 6 of each heart valve leaflet, and we were able to produce these graphs right here. And you'll notice that the tricuspid valve leaflets were actually tested to half of the pressure of the mitral valve leaflets based on their estimated physiological loading. But you'll notice that the radial direction was consistently longer than the circumferential direction. So we found this anisotropy in the tissue, and on average that was 30.7% higher, just noting that peak stretch value. We also found nonlinearity where the leaflets would actually stiffen as you approach that physiological load. And initially they have this toe region, you can see this low region right here, and then you get this very stiff region. And that was pretty characteristic of all the leaflets. And finally we found these preconditioning stretches where the tissue actually deformed a fairly significant amount between our loading the tissue and the end of that preconditioning cycle where we conditioned it and returned it to its in vivo configuration. So we also found some actually similarities between the peak stretches of the mitral valve leaflets. So there wasn't a ton of difference here between MVAL and MVPL in terms of peak stretches. So you can see kind of small difference there. Whereas in contrast, we saw a lot of difference in the peak stretches of the tricuspid valve leaflets. So you can see over there pretty significant variance. And basically what we're finding is that these leaflets need to be treated on an individual basis because they have these differing material properties. And that leads us actually to the constitutive model development. So what we're going to do is take all this experimental data that we've obtained and then fit a model that would allow us to use this in computational models later on and for multiple purposes. So we had to make some assumptions for this, like in any model. And one of the first that we made was that this was a pseudo-hyperelastic material. So this allows us to develop energy density function for the tissue based on how it is deformed. So basically relate the strain to energy developed in the tissue, and then you can actually compute a form for the stress versus strain. It's a fairly standard procedure. We also assumed this was a transversely isotropic material. This was based on an assumption made in a paper by Dr. Lee and others, and this actually produced pretty valid results for fitting of leaflet mechanical responses. And in that assumption, we actually looked at the microstructure of the material and related the mechanics of the material to the microstructure through an inverse method. And we also assumed that incompressible material, this was very standard, and that's based on high water content within the tissue as well as other reasons. And this led us to the form shown here. So there are actually 4 principal components of this strain energy density function, and so we'll work through them individually. This first component you can see is the isotropic energy of the non-fibrous leaflet components. So there's no directional dependence here, and this is actually most relevant at low stresses. To understand the next 2 terms, it's important to note that the heart valve leaflets are actually enforced by this matrix of collagen fibers that runs throughout them, and these fibers have a very varied orientation throughout this, spatially throughout the leaflet as well as throughout the depth. But you'll see that this first term corresponds to an exponential isotropic response of the fibers. So this is considering that delta parameter is a fiber spread, and this is considering a fiber spread of 1, which is totally spread out, no preferred orientation. Does that kind of make sense? And so that exponential term is the exponential response of the collagen fibers themselves. They've been tested and are known to have that exponential response. And we also have a term here to capture if there was a very aligned set of fibers. So generally your delta is actually going to fall somewhere in between those two, and both of these 2 terms are going to play a role. But this second term is a very anisotropic exponential response to account for those very aligned fibers. And finally we have this penalty term, which enforces tissue incompressibility. And we're actually able to solve for this p-value through analytical methods. So we're left with 5 parameters, and the question of how to fit those parameters is what I'll address right now. So here's our 5-point parameters, C10, C0, C1, C2, and delta. So we initialize that fitting parameter set, and then we basically establish this current parameter set, and that'll allow us to iterate through it. So from there, we pull in our testing data and compute this residual value. And so what this is basically how well the model with the current parameters fits our data. And so if that residual is high, that means that we don't have a very good fit, residual drops, and our fit's getting better. And we ask this question, is R less than 10 to the negative 6? That's what we call our tolerance. And generally for that initial parameter set, it won't. So we'll have to iterate a couple times. What we do is we use this differential evolution algorithm to generate a set of mutated parameters. I'll call that beta mutated, right? And that kind of is symbolic of evolution in the population. So those mutations can kind of be randomly occurring. And then we check whether this mutated set has a lower residual than the original set. And we take the best one of those and we loop through this again and again and again. And those mutations allow us to find that optimal set of parameters. And so after basically 1 looping, we'll also check this condition right here. And if either of these conditions are true, then we know that we've gotten to our optimal set. And this basically just comparing the set of parameters that's currently used to the prior set of parameters used, and seeing if the difference between them is basically negligible. Because we don't want to loop forever, so that kind of helps us to break out of that loop if we just are approaching a converging solution. And then once we get a yes for one of those conditions, we get our accepted parameter set beta optimal, and we do that for all 5 heart valve leaflets because, as we noted, we have different responses between them. And this actually led us, so here's 2 examples of our model fits right here. You can see how they actually did quite well. We have the equal biaxial loading ratios shown. We've also been working on the non-equal biaxial loading ratios currently. And actually there's the table for all of the parameters that you can see here. There's a couple interesting results of that. Remember this C10 was the isotropic component at low stresses, and that basically accounted for that linear isotropic response. And so you'll see how that was almost negligible on the mitral valve leaflets. We saw a very small linear region, and really all of this nearly flat behavior could be accounted for by the exponential terms. Whereas for the tricuspid valve leaflets, we saw a much larger toe region, and so we saw a much larger linear effect at the beginning, and that's why we get a larger parameter there. And there's these kinds of patterns throughout. So our fits, we're actually pretty happy with our fitting for the data, and this moves us to kind of the summary of what we've done in this study and the impacts of it. So basically throughout this study, we have shown that all of these atrioventricular valve leaflets exhibit this mechanical anisotropy, nonlinearity, and preconditioning stretches. This had been not previously shown for the tricuspid valve leaflets. We showed that they required a consideration on an individual basis because of those differing mechanical responses. We also showed they can be modeled as transversely isotropic materials with considerations for their microstructure. So as far as the impacts of this study itself, this is hopefully going to allow us to move one step closer toward computational modeling, and specifically patient-specific computational modeling, to help inform that surgical practice. So this better understanding of heart valve leaflets can be incorporated into these computational models, and hopefully with patient-specific geometry. And with that, we expect to be able to provide a valuable tool to surgeons to get a better understanding of what's going on and how potential treatments would impact that patient. We also hope that this will inform biomaterial selection. A lot of times these biomaterials are actually isotropic, and so we see a lot of non-parallels between what you're replacing the valve with and how the valve behaves in the native state. And so we hope that through this thorough characterization, we can kind of get a better idea of how to better design these valve replacements. So I'd like to acknowledge my thesis advisor, Dr. Chantal Lee, the Office of Undergraduate Research for sponsoring me to come here, and just the help and funding from my home department, Mechanical and Aerospace Engineering. So thank you for your attention. And yeah.
So we have a couple minutes we can take for questions because we lost a talk, so we can be flexible. Sorry, sir.
So when you dissect the heart valves, do you do anything to take into account that, I'm assuming that there's probably some residual stretches in them in vivo. Have you taken that into account yet, or is that
Yeah, so
planning on looking at?
So yeah, there have been a lot of studies on like residual stresses, and yeah, they have been found in vivo. So what we try to do that to account for that is use our preconditioning protocol to return it to kind of that in vivo state. But we realize that that's not perfect, and since we're only looking at that small section of the leaflet, there might be kind of some pre-stresses that are lost as a result of that cutting procedure. But it's pretty standard practice and has been used for aortic valve leaflets frequently, and other valves.
I think you mentioned you used I think it was called a differential evolution algorithm. Is that at all similar to a genetic algorithm?
Yes.
Okay.
Yeah.
Gotcha. Because I just didn't recognize it. Thanks.
I think that will be covered a little more in depth later on.
Gotcha. Yeah.
Quick one myself, actually. So why? It seemed like there was more variability in the radial direction when you were showing the initial results plots. I was wondering if you know why, or had some thoughts on why that might be.
So a lot of that might be able to be attributed just because that radial direction response is actually just so much higher than the circumferential direction response, and so percentage-wise, the difference might not be as substantial. I think we kind of found that in our results. But since the circumferential direction was lower, you can't see that absolute difference as clearly, but the percent differences were comparable, I think.
Well, let's thank Sam one more time.